Um, hey everybody, Kris Fobes here. As Amy was saying, that one of the founding members of PolyBio, and also founder of Gene Savvy, which is a genetic testing company. And I just wanted, my presentation is probably going to be a bit less scientific than the rest, but I get a lot of questions about RCCX and specifically its role kind of in these persistent pathogen situations. And so I just wanted to kind of go over a couple of things and theories and ideas that have come across my table, and hope that it kind of sparks conversation or thought, at least in the direction of RCCX and the roles it might play in these clinical situations. So let's see, move this whole thing. Okay. So first, what is RCCX? RCCX is a cluster of genes found chromosome 6, HLA class 3. This is what it's considered. And RCCX just stands for the genes that are kind of involved. So the RP1, which is actually more commonly known as STK19. There's another gene that's listed if you look up RP1, and it's not as associated. So I would stick with the STK19 idea. The first C is C4A or C4B. That one's probably one of the more known ones. The second C is the CYP21A2. And the X is from the TNXB. And down here at the bottom, you can kind of see how this is laid out on the chromosome. So you got the RP1, C4A, and B. There's HERVK here, which I'll discuss a little bit later. Then we got the CYP21A2 and TNXB module. So we'll start with STK19, which a lot of people, aren't too sure about the role of STK19 in this. And so I was doing some research, and this is at least what I came across, and the way that I think it plays a role in persistent pathogens is that it has this, it's known to mediate this NRAS signaling, which mostly is enhancing the binding to downstream effectors. And where I thought that was like the biggest role in this is that these downstream effectors, sorry, the scene is right in the way for me. Anyways, NRAS is this viral oncogene homologue. So that plays a role in the viral genome being transcribed in the DNA genome of the host via reverse transcriptome. So that's where we start seeing something like retroviruses, or I think where we start seeing these persistent pathogens where they continue to replicate even after the acute phase that we think that they're responsible for. The other thing that this might play a role in is this DNA provirus hypothesis, which I won't go into too much, but it's basically just talking about how viral RNA can be transcribed into our human DNA for ongoing replication or transcription. So it's a pretty interesting thing to look into. C4A, obviously this complement component 4 is very heavily involved in the complement cascade, which is our immune response, is a big part of it. Complement component 4 comes in kind of two different forms, the C4A and C4B. I won't get too much into the different blood group types and whatnot, but there's just two forms. C4A mostly involved in the classical activation of the complement cascade, which deals a lot with the management of antigen antibody immune responses, which is obviously a big topic in something like COVID or any kind of persistent pathogen really, or autoinflammatory disease or anything like that. C4A also mediates inflammatory response. So something like a massive or, cytokine storm or histamine release, anything like that, can also be associated with C4. So it's a pretty big role there. And then one of the biggest things they get questions on is this C4A long versus C4A short, which has the C4A long version includes the reverse transcript of HERV-K in its ninth intron, versus the C4A short, which doesn't include the transcript for HERV-K. So, I think that this is one of the ways that we can see increased HERV-K expression in response to some of these pathogens or infections that we get. CYP21A2 is a P450 enzyme. Obviously, those are really heavily known to be involved in drug metabolism. This one specifically also has a lot of roles in lipid metabolism, in adrenal, efficiencies and steroid processing, things like that. So, see, I wrote down here, reduced CYP21A2 contributes to adrenal inefficiencies, which, any of these inefficiencies in those in adrenals is going to increase risk of, possible viral infections and reduce your ability to manage the stress or dehydration that could be a response to an infection, especially something like COVID, where we see a lot of blood coagulation issues. Dehydration could play a major role in those. The last one is this TNXB, which is usually associated with something like Ehlers-Danlos or some kind of tissue disorder. But the flip side of that could also accelerate this collagen response or fibrotic response to damage. We see a lot of like scarring tissue and stuff in the lungs, and that has been associated or excessively associated to TNXB overexpression. So that is something to watch out for also. Another big question I get about RCCX are the different types of genomic anomalies that we see. So obviously I talked about C4A short and long. It's the retroviral insertion in the ninth intron of C4A. That's something we look for. RCCX modules and copy number variants. So this is the genes within the RCCX module kind of duplicating on the chromosome. I will show you a little image of that later. And the most common RCCX module arrangements that we see are these monomodular and bimodular and trimodular arrangements, which is just duplications of basically the C4 and the CYP genes within this, like the middle of the modules. And then obviously we also see single nucleotide variants within the module all over the place. And the SNVs and SNPs can, affect gene function or expression or any of those things. And those can be commonly seen. We can find rare ones, and they all kind of have these slight different effects on, like I said, the function of the gene, and activity, and possibly the transcription or expression of those genes that they are associated to. One thing that is brought up quite a bit with SNVs is the ability to determine whether or not the SNV is actually in the gene or whether or not it's in one of the alt genes or pseudogenes that are commonly mistaken for the actual genes of RCCX. And so, something like a SNP or SNV that pops up in ancestry type testing, it would be hard to tell if it's a variant that's actually in the gene or if it's in one of the pseudogenes associated. So some, higher end sequencing or analysis that can determine that between the two would be required for, finding that out. Here's an example of some of the more common module arrangements we see. You can see they all start with the STK19 or RP1 gene, and then the duplicates happen in the C4A and CYPs. And then we also have this ability for those pseudogenes or alt genes to kind of come in here also and be part of those modular copies. So that's where we'll see that. This was a study that we did to look at RCCX patterns in healthy controls. And what we saw, at least in this little group that we did, was kind of this underrepresentation of HERV-K and excessive amounts of C4 or RCCX copies. So that was pretty interesting to see that, at least in these healthy controls that we did, we weren't seeing excessive like bimodular, trimodular RCCX copies, and we also weren't seeing HERV-K insertions within C4. So that's what this is kind of showing. And so just some key takeaways real quick of kind of what we went over is that STK19 or RP1, it might support viral integration in the host genome or retroviral trans activation. So I think that that's actually a bigger player than most people think about. Obviously, it deserves a lot more research and support around the theory of that. I hope that with PolyBio we can start making progress that way. But I think viral integration into the host genome is going to be a big player. C4A, obviously, it's plays a big role in our immune response and antibody antigen reactions. Reduced C4A activity can lead to pathogen persistence via, just not being able to clear the pathogens. Excessive activation can be a contributor to an overactive inflammatory response, like a cytokine storm. So we obviously have issues with that also in COVID. And then C4A also acts as the host for HERV-K, which I know that Amy has talked many times about associations between increased HERV-K expression and persistent pathogens or infections. So I think that's going to be pretty interesting to see. CYP21A2 can lead to adrenal inefficiencies, increased risk of viral infection, and lower ability to deal with it, deal with the stresses and also dehydration that could be associated. And then last but not least, the TNXB stuff. Increased TNXB has been researched and associated with increased risk for pulmonary fibrosis, which is something we commonly see in these COVID cases. And I think it's going to be one of those long-term effects that are going to be in a response to COVID. And then decreased expression has been, associated with collagen or tissue hypermobility and EDS. And I think that that could play into maybe enhanced tissue degradation or even something like blood-brain barrier dysfunction if we looked into that a little bit more too. So I know mine was quick and simple, but I wasn't sure what all to put in there and whatnot. So that's kind of it. I wanted to thank Sharon Legatheri. She did a ton of research and she's really brought awareness to RCCX So I just wanted to point that out and point to her website too. She she has a lot of great information on how it relates to illness and clinical situations.
Thanks,
So
Kris. So I wanted to ask you a question. So for example, in schizophrenia, there's these big genome-wide association studies where, 108 risk genes pop up. And the main hit in these large, 100,000 subject studies is right there in chromosome 6 in the major histocompatibility complex, which is right on the CD4 site. So it's been known for a while that CD4A versus B and long and short forms can predict risk for schizophrenia. But SECAR in 2016, I guess, showed that the presence or absence of a HERV in that site in CD4A actually predicted the amount of protein produced by CD4A gene above and beyond the genotype. So CD4A is one of the important, immune modulators that tags synapses for pruning during development. So the thought is that maybe during late adolescence, when that pruning happens, it goes too far in schizophrenia and like the dorsolateral prefrontal cortex layer
yeah,
3 and stuff. So I'm
actually,
curious, are there any other sort of known downstream effects of presence or absence of HERV in that site?
you, boy, I've seen quite a bit, and actually, synaptic overpruning is something that I really got passionate about there for a bit in a case that I worked on where like schizophrenic type of symptoms were present. But as far as the HERV, there has one thing that I actually thought was pretty interesting, and I explored a little bit, and I need to or want to explore some more is um, the idea that HERV also might provide some kind of protective role against some of these immune responses or some of these viral responses that we get. There's a lot of research that kind of points against it, but there's some that points for it. And I think it's something that's going to be interesting to look into. Um, yeah, I think that there's all sorts of things that we could see as a downstream effect of HERV expression, specifically from C4A in this uh, major histocompatibility complex section.
Kris,
So
yeah. Oh no, go ahead.
no, you go ahead.
No, it's okay. Well, Kris, and how did you I realized this other doctor started with RCCX, but why did you become interested in it? Did you thought it was going to be a module that was going to impact like some of the chronic conditions you were working on?
Yeah, it was a combination of a couple of things. Obviously, there's a lot of topic happening around it right now, and the specific case that got me started on it was actually more involved in CCI than it was with COVID. And so we were looking at how RCCX could contribute to CCI and like Mike was saying, these possible schizophrenic-like symptoms. And so TNXB and its relation to EDS came into play, and then also synaptic overpruning or synaptic pruning in general. Those two things came to mind, and it just pointed directly to RCCX. And it's as soon as I started looking into it, it was just kind of really interesting, and there really just wasn't that many tools for identifying or working with it, and I found it fascinating. So
Cool.
So, Kris, one of the things that I'm curious about is, I always sort of thought that the HERV placements were somewhat random, but I guess that's not really true. There are predictable sites where they're sort of known to be in people. So, I guess two questions. One um, is it the case that there are some known places where they have, possibly clinical effects at other sites? And then two, does standard genetic testing get at the presence or absence of HERV, or do you have to do something special?
oh, yeah, so those are actually really good questions. One is the first one about known locations of possible HERV-K that could have clinical effect, that would absolutely be true, because something like HERV-K insertion into C4, um, could have a much different clinical impact compared to location in something else, just because the idea is that at some point, we transcribe that HERV-K sequence at the same time that we transcribe for C4A. And so if something gets expressed or something drives expression of C4A, it could drive the expression of HERV-K versus HERV-K located in a different section or somewhere that's not maybe in part of our immune or inflammatory response, right? So that definitely is
That makes sense.
a factor. And then as far as genetic testing goes, because HERV-K can be located at different points throughout the chromosome, when we look for HERV-K specifically, we narrow down the HERV-K reads to to specifically the C4A locations on the chromosome, so that we don't get HERV-K reads from different sections that we're not looking for, because it can be all throughout the chromosomes in different sections, in different genes, but we're specifically looking for this one.